Okay. I think we're good to go. All right, so it is my great pleasure to introduce tonight's guest, Dr. Lindsey Swierk from the State University of New York at Binghamton. For those of you that were with us in the Amazon in 2019, you had the pleasure of meeting Lindsey in first person, and maybe even potentially got to help her lasso some lizards with her cool little lizard lassoing equipment up at the Canopy Walkway. So we're just thrilled that she's on our team now, and I can hardly wait to get back to the Amazon with her in 2022. So we're working on plans for that as we speak. For those of you who haven't met her yet, Lindsey is a research professor, again, at the State University of New York in Binghamton, and her specialty is behavior and ecology of species in a changing world. She focuses primarily on reproductive ecology, sexual selection, and animal communication, with a specific focus on lizards and frogs, one of which is near and dear to all of our hearts, the shovel-tailed lizard at the Axe Canopy Walkway. What I love about Lindsey is her work is so deeply grounded in natural history, which I think is just an incredibly important teaching tool and storytelling tool, and it's the essence of field biology. And she's doing her work with an emphasis on conservation and human impact in the northeastern United States, as well as in the neotropics. She's heading down to Costa Rica in a few short weeks. And then she also works with us in the Peruvian Amazon. So we're just delighted to have her with us tonight to share her work with lizards and, you know, some of her stories about being a woman field biologist and ecologist, and what that looks like. So Lindsey, I'm going to let you take it away, and have fun, everybody. I'll see you at the end.
Thank you, Krista. Can everybody hear me okay? All right, well, I will assume that you can, unless someone waves frantically at me that you can't. Krista, thank you for that wonderful intro. And thank you, Seth, for the thumbs up. I'm really, really happy to be here. The webinar series and just seeing all of you has really been a silver lining in this kind of dismal past year. As Krista said, for those of you who were with us in 2019, you might remember me. I was the weirdo who was always kind of lurking out underneath the lodges, looking for lizards underneath and up on the Canopy Walkway. And I was also the one begging everyone to tell me if they saw any lizard anywhere at any time. So although I'm going to present this tonight as in a seminar format, I really encourage you to please interrupt me if there's something that you have a question about or you want to chat about. I'd really rather make this more of a conversation. Unfortunately, despite teaching on Zoom all last semester, I still can't figure out how to share my screen and view the chat. So if someone can just let me know if there's a question in the chat, that would be great.
I've got the chat monitoring going on, so I'll let you know.
Excellent, thanks. Okay. So first, I'd like to try to remind you all of the true sense of wow, that sense of wonder that we had when we were all down there in the Amazon. In this year of kind of no field experience for me, no field discovery, which is really integral to my job, what I love doing, remembering what the experiences we had and everything, all the cool critters we found, has really kept me motivated during this weird year. And I'm sure a lot of you are very much the same way. As Krista said, I am a behavioral ecologist, which means that I have the pleasure of exploring animal behavior in light of evolution and ecology. And I've chosen this slide to start on with its really cool double rainbow that was taken from the Canopy Walkway, because a lot of what I do in terms of behavioral ecology is focusing on animal communication, and in particular, how animals communicate using color and visual signals. So a lot of what I do looks at color across the spectrum. Okay, so tonight my plan is to give you a little general background about herps, just to give you the general context. And then I want to talk a little bit about how animals see the world and how we can kind of get into their heads in order to better understand their natural history. In order to understand their natural history, though, we have to first acknowledge our own biases from just our human perspective. You know, we are humans, so how we perceive the world as humans, and also biases that have arisen based on the societies that we live in. Okay, so herpetology is the study of reptiles and amphibians, and I have to say I think this group is definitely the weirdest and sometimes the most misunderstood taxon in the animal kingdom, and maybe that's why I love studying them so much. They are hugely diverse. We have some general similarities in the two groups. Obviously, amphibians have to have a life stage that's at least partially in water. Reptiles do not. But we see similarities between these two. We see the general tetrapod lizard-shaped body in both reptiles and amphibians, the salamanders versus lizards. We see the snake body shape repeating itself in both reptiles and amphibians. So we have the snakes, we have the legless lizards. In amphibians, we have these weird looking things called caecilians that look like amphibian snakes. But the important thing to recognize here is that even when we see these similarities between the two groups, this is an example of convergent evolution. Herps as they have evolved are really kind of a grab bag. They're all mixed up. So in terms of amphibians, we have frogs and salamanders, as everyone knows, those weird caecilians that I was telling you about, and then a bunch of extinct clades like the bifetids, which are essentially amphibian crocodiles. Weird, I wish I could have seen one. And then the reptiles, of course, we have lizards, turtles, crocodiles, snakes are grouped in with lizards. We also have something called tuataras, and this is a group with only one species remaining in it, and they're lizard-like things found in New Zealand, live over 100 years. Again, really bizarre and cool. Okay, but the point is the fact that we call these things together herps is really just a historical legacy, and it's kind of just a grouping of convenience. Herpetologists and non-herpetologists alike kind of take issue with this. Most generously, we might call it a group of convenience because there are some similarities in the way we study both amphibians and reptiles. Least generously, we might call it a mess, but either way, it is true. They don't really belong together, but that's how it is. All right, herps in the ecosystem can act as everything from little chicken nuggets in the forest, feeding all the other species, or they can be predators themselves, cleaning up our pests. Sometimes there are really complex interactions. Sometimes the hawk eats you, sometimes you eat the hawk, like this poor roadside hawk getting eaten by the boa in Brazil. And herps can also—
I'm just going to say wow on that one. That's amazing.
I know, right? Isn't that crazy? All right. So herps can also be voracious herbivores and therefore also be ecosystem engineers. So I did a little work in the Caribbean on iguanas, and iguanas that have invaded some Caribbean islands like Puerto Rico have actually completely changed the forest structure because of their voracious defoliation of the mangroves. So they've changed the structure of the mangrove forest, and also, frankly, they poop a lot. So they change the communities that are in the soil. So the point is that herps serve a hugely diverse role in the ecosystem, and I can give 5 talks describing the roles that they have. But in the interest of time, we'll move on. And I'd like to highlight also that worldwide, the greatest diversity of herps tends to indeed be in the tropics. Reptiles are actually the most speciose group of tetrapod vertebrates, and by that I just mean that there's the most species within that group. There's actually 12-ish thousand, maybe more now, reptile species worldwide, and over 500, probably close to 1,000 in the Amazon, but of course we really don't know yet. Amphibians are catching up. In the past 30 or 40 years, we've increased the number of amphibians that we've identified to science by 60%. And in part, that's largely due to Peru. So you can see that just in 2017, Peru and I guess Madagascar were leading the world in identifying new amphibians. So there's 1,000 species, probably more of amphibians in the Amazon, and that's of the 9,000 species there are worldwide. Okay, so now for the one sad note that we all have to pay due to. Unfortunately, there is a race to identifying all of these species to science before they go extinct. The rate of extinction is accelerating for both reptiles and amphibians. And many of you know these reasons already, but I'll just rehash the list. We've got habitat loss topping the list as the #1 cause of extinction for both amphibians and reptiles. We've got invasive species, we've got pollution, which amphibians are especially sensitive to, right? Disease, climate change, and we also have the kind of unique, I won't say totally unique, but sort of unique problem with reptiles that they just generally have a bad rap for no apparent reason. People are fundamentally trying to get rid of snakes from their property every way they can. There are rattlesnake roundups, poisoning. But then on the flip side, we also have reptiles and amphibians being popular as pets because they do tend to be beautiful creatures that are, in some cases, relatively easy to keep. So 12 years ago now, I can't believe it's that long, when I first went down to the Amazon with Steve, we were looking at some of the creatures that were for sale as meat items in the Baleine market. And 12 years ago, I took this picture of an Amazon river turtle. And there were, dozens of these things just in the one day that I was there that were for sale. And then also on the medicinal side of things, there were turtle fat products throughout the market as well. Thankfully, when I was there in 2019, I don't think I saw any river turtles, and that is largely due to community-based conservation efforts. So there is some hope. Okay, but the thing that always drives me is that any kind of conservation efforts that we want to initiate always have to start fundamentally with natural history. And natural history is simply the art of observing nature, right? It's what we do as kids. These are my kids. And, a few months ago, well, not a few months, last summer, I guess, we were poking around a stream and looking for tadpoles and things. But that very fundamental thing that we used to do as kids is the very thing that we rely on in ecology and evolutionary biology to found our studies on how animals relate to the rest of the ecosystem, how we can conserve them, and their history on this world. So, it's really important that we understand natural history not only for conservation, but we also have reasons related to the optimization of human-wildlife interactions. We have lots of examples of bio-inspired designs, right? Really cool, funky things that make our lives better based on the structures that animals show. And also human health benefits and agriculture as well. Unfortunately, in let's say the 20th century, emphasis on natural history has declined, and that's largely because there's no money in natural history. It's time consuming, it's expensive, and if people can get more papers out and more grants out without putting in a lot of time just observing nature, that sadly, is just the trend that we're seeing. The next generation of scientists, though, we are fighting back, so we're going to see how it goes. But the bottom line here is that we need natural history for conservation, and in order to have effective natural history information, we need to look through the species' eyes. We need to see what they're seeing, and that is hard because we are people as humans that are inherently full of biases, right? We have both biological biases, as I mentioned earlier, where we as humans see certain colors, we hear certain sounds, and there are also social biases related to the societies that we were raised in. So right now I'm going to start to chat about those biological biases and how we as scientists try to look through really objective lenses when we're studying animals and their natural history.
Hey, Lindsey, can I interject a question here that came up that's kind of a good time to ask that? Christy is asking, what do you think about the hunting of snakes that are invasive species now in the United States, since you're talking about biases?
Well, that's pretty controversial. Personally, I support it. But I also will say that a lot of money is often spent on futile efforts. So it really depends on the case. I think I know the cases that you're talking about, and generally I support those if it's done humanely, but I agree it is pretty controversial. Are there any other questions in the chat?
Not yet. Thank you.
Yep. Okay. So whenever I teach this to my students, I always try to turn it on its head, and instead of trying to get others into the heads of the animals, I try to flip it on its head and have people think what it would be like if animals were studying us, right? And frankly, if animals were studying us, they would really think that we were completely blind and completely deaf, because there are so many stimuli in the world that we simply don't or can't respond to. Responding to magnetic fields, like many migrating sea turtles do, or all migrating sea turtles do, and detecting the direction of polarized light. And this is kind of a fun tidbit I like to slide in my classes, because although we think that we can't see the direction of polarized light just because it's not something that's familiar to us, we do have a relic ability to do so. So if you stare up at a sunny sky, or if you look at a white LCD screen and kind of turn your head back and forth really quickly, you see these things called Heidinger's brushes, and that indicates the polarity of the light that you're looking at. And usually my students at this point immediately pull up a white screen, but it's kind of a cool trick. Anyway, one thing that we absolutely don't have any evidence that we can currently sense is electroreception, as many salamanders can perceive. Also, there's infrasound, which is sound that's too low for us to hear. And animals like alligators will use infrasound in their mating calls. And I've heard that if you listen to this for a really long time, it'll give you a headache, even though you can't actually hear something, because it's just so loud for so long. Okay, also there are many animals, including snakes, that can perceive infrared radiation, and boas and vipers and pythons can do this with these little heat sensing pits on their face, and that's so that they can detect mammalian prey, which are making their own heat. And then of course we have ultraviolet vision. And the picture here on the right is the lizard view of the same scene that's on the left, which is what we see. And on the right, we can kind of see how it's false colored, of course, but brighter areas of bright blue on those lizards. And that is, again, light that we just can't see because the wavelength is too low for our eyes to pick up. Okay, so kind of the reason that I'm telling you all of this is that we live in an information-packed world, and we have biased perception out of necessity, just to allow us to make sense of things. This again is that Sicilian. It's a neotropical amphibian. It lives underground, sometimes underwater, depending on the species. And it only responds, its entire world is filled with three things: vibration, vibration, smell, and taste. Doesn't really see, doesn't really hear. So if you kind of got into the head of this Sicilian, then it would be completely foreign to anything we've ever experienced, right? But it is a rich and information-packed world for the Sicilian. So we've borrowed this term from philosophy, which is called Umwelt. And it's this idea that each animal has its own little world that it responds to. And it has a world that is built on its own sensory capabilities. Now, this is important because moving forward, I'm going to tell you about a bunch of lizards from the Amazon and how they communicate or potentially communicate. And I would like you guys to keep in mind the idea that the evolution of all signals has to come from the sender. That would be, the lizard bearing the color, the sender's own umvelt, and the umvelt of the receiver. Okay, any questions, Krista?
Nope, not yet.
Cool, okay. All right, so now I'd like to take you back to the path between Napo and X and try to remember what it felt like. It was in some places very dim, right? There was not a lot of light shining through the forest canopy. It was very warm, sometimes very still as well, without much wind. If you were a little creature running around on the forest floor here, you would have an umvelt that had very low levels of light, lots of places to hide, very warm, very still, to some extent. Now contrast that with the umvelt of a lizard on the canopy walkway. Okay, a lizard on the canopy walkway has an abundance of light, and that light is completely full spectrum, contrasting with the green and the yellow light that's underneath the forest canopy. So the lizard on the canopy, though, has to deal with visual noise, which is kind of just motion, right? If there's a little bit of wind, a little breeze, you've got that visual motion. You also have a lot of glare from the sun. So the umvelts are really completely different here. Okay, so at this point, we're gonna walk through, I'm gonna remind you of some of the lizards that we met while we were in the Amazon. And I'd like you to, again, please stop me if you just want to chat about the lizards in general, because there's so much we could talk about regarding lizards. But I'm going to try to illustrate how the coloration of the lizards relates to the habitats that they're in. So our first subject is one that is kind of the poster child of the canopy walkway. And you often could hear this one before you saw it because you could hear them skittering up and down the trees. And right there, I'm not sure if you can see my arrow or not, but right there is a tree hole, which is kind of like the front doorstep of the homes of these guys. Can anybody remember who they were? I gotta get the chat up. No chat. Well, there we are. All right,
Nothing yet. Go ahead. Did anybody know who lives in that hole? That little hidey hole? I do, but that'd be cheating.
Well, who is it, Krista?
It's the shovel-tailed lizards, right?
Yeah, absolutely. This guy, right? Okay. So shovel-tailed lizards, also called the Amazon thornytail, probably so named because they have these kind of flat, spiky tails. I have a soft spot for critters that people don't know much about. So I have decided that, moving forward, this will be one of the species that I focus my efforts on. One of the reasons, perhaps, that they're understudied, and when I tell you they're understudied, they are crazy understudied. There are literally 3 publications on this species compared to hundreds that there are for lizards that live on the forest floor. Thousands that live on the forest floor. This guy's got 3. Perhaps one of the reasons is simply because they're really hard to get to and they're really hard to capture. So finally, when we managed to capture our first male, everybody had to pose with him. And this is a very handsome adult, sexually mature male. And this is a lady, his counterpart. So the females and the juveniles look very similar. They mostly have a diet of ants. And one of the morpho faculty actually told me that they've seen them eating bullet ants, which blew my mind, but they've got these big chunky heads. So if anybody's going to crush a bullet ant, I would say these guys would. And they also have the reputation, at least, of living in social groups of a dominant male, a subordinate male, a bunch of females, and a bunch of kids. Now, I say the reputation because the one paper that has studied this species is a paper that was based on the felling of one single tree alone in a field. And then the herpetologist found all these lizards spilling out of the tree. So take from that what you will, but there's a potential that these could be socially living, which would be very interesting in the lizard world. Okay.
And I'm sorry, I just have to say this out loud. I think most of us that go there frequently take this for granted that we're going to see these guys. I honestly had no idea that there are only 3 papers, like period, on these lizards. So every time we see them up there, this just like gives me goosebumps. We could be seeing like brand new behavior. So I'm just going to throw that out there, folks, that you may have watched lizard behavior that nobody's recorded before and didn't even know it. So yeah, that's all.
That's a really good point, Kristen. No, that's absolutely totally true. And you know, I'm going to actually, in the next few slides, I'll show you some other lizards that people know almost as little about. And there are some pretty surprising things that, you know, we've uncovered in we were there 10 days looking at these guys, and I mean rather other species that it's really astounding. Okay, so catching these guys herping in a canopy is a complete struggle. So this is Peter, one of Steve's undergrad assistants from this past summer. And, you know, everything we did during those 10 days was mostly just pilot data. We were getting familiar with the techniques, figuring out what worked, what didn't. So as I mentioned, these guys tend to spend the nights in tree holes. So we figured that one easy way of capturing them could be by placing these minnow traps with very firmly secured to these tree holes in the evenings once the lizards, the shoveltails, were in the tree holes already. And then the next morning when they came out, they would be in the minnow traps. This did not work at all. We were really hoping, but you know, I don't know whether it was just that we didn't have enough days to try it out, or we were putting them on at the wrong time of day, but it just didn't work. So again, trial and error. One thing that did work is lassoing lizards. And lassoing lizards is kind of just a fancy way of saying you stick a slip knot on the end of a fishing pole. And this was a nice extendable fishing pole that we could pack in our suitcase. And you put that slip knot gently around the lizard's neck, tighten it when the lizard pulls away, and then you can kind of easily lift the lizard off the tree and you know, capture the lizard at that point. As a complete side note, I just thought it was interesting that many of you may have heard the term noosing a lizard before rather than lassoing a lizard. And noosing a lizard is a term that we as a herpetological community have decided to really move away from because it is so charged with really negative associations for a lot of people. So you may have heard noosing and lassoing, noosing instead of lassoing, but they're effectively the same thing. Okay, so this was another one of Steve's undergrads that we worked with. Her name was Lauren, and she was a really expert lizard lassoer by the end of only a few days. As I said, this worked tolerably better than those the minnow traps that we put on the tree holes. We also tested out some glue traps in its pilot stage. So, you know, this is again the process that we go through to figure out how to work with a new species that's extremely hard to access, because as you can imagine, if the lizard just moved 2 or 3 inches away from the end of our pole, which they often do, you can't get them and you have to walk away while the lizard's sitting there taunting you. Okay, so once we have captured these guys, and we did get a nice handful last time we were there, here's a female on the left and a male on the right. One thing we wanted to look at was why they have the colors they have. So I mentioned, you know, females have the same color more or less as juveniles. It's kind of this the speckled green pattern, and males have the bright red head and the black jumpsuit on. Anybody want to take a guess why juveniles and females look similar, or subordinate males and females look similar? Yeah, it's really a beautiful green, isn't it? I think we underestimate the female coloration because everybody's always so amazed at the beautiful male coloration.
Seems like camouflage, like, right?
Exactly. Yeah. Well, possibly. But you know, it's a good guess. So the juvenile males and the females are similarly colored, basically, so the dominant males don't end up beating up on the subordinate or the juvenile males, and then the juvenile males can feed and grow, and eventually once they're big, they'll show dominant colors and then they're ready to fight. But before then, it's kind of just they'll hang around. If they mate with females, they'll sneak mate. So they're kind of wearing a disguise. Okay, but we wanted to know why these two these two sexes were different, but also I wanted to know why the male in particular wore these colors, why the red head, why the black body. So in order to do that, I needed to make sure that my own perception of lizard colors wasn't biased by me being human and me looking through human eyes. So I used this gadget that many of you have seen me use already called a spectrophotometer. And this is a field portable one, which we built custom just so I could take it up into the trees and into the jungle. And it's basically, the best way to describe it, I think, is an artificial eye. So it shoots out light, which I acknowledge real eyes don't do, but this gadget will shoot out light of a known wavelength, and then it'll measure how much light of the different wavelengths will come back. So when I say wavelength, I just mean color. So it shoots out light of known colors and then measures how many of those colors are reflected back. When light is reflected back, that's in essence the color something is. So as an example, when we're looking at the shoveltail's red head, we can get a spectrum that looks like this. And I'll explain it very, very briefly. I'm always happy to chat people other times if they're interested. But the horizontal axis here is the part of the electromagnetic spectrum that we can see, about 400 to 700 nanometers. Below 400, those are UV colors, and above 700, those are infrared colors. Okay, so other animals can see that, we cannot. On the vertical axis, though, that's the amount of reflectance. Basically, so the curve is the amount of reflectance at those different wavelengths, the amount of color that is showing up at each of those particular wavelengths. So I can confirm, looking at this spectrum, that because the peak is right here, around 690, that the lizard's head is indeed red. So good, my eyes haven't fooled me. But the other question is, why is the lizard's entire body not red? If it's good to be red, because red in lizards is a way that they can show you, basically it's how much carotenoid that they can accumulate, and they can show you how good of a fighter they are, or how good of how good their immune system is, various things. Why is the lizard's whole body not red? Why do we have this little black jumpsuit on? Anybody want to take a guess?
Guess away, folks, guess away.
Predators, special selection. Excellent. Definitely heat. Yeah, I was hoping somebody would say that. Absolutely. So what is the color you don't want to wear when you're sunbathing and it's already hot out? You don't want to wear black, because then all of those rays just absorb into you and you get really, really hot, and it's generally unpleasant. Well, these guys who are ectotherms, which means that they have to use the environment to control their body temperatures, in the morning when they wake up, they're pretty cold for lizards. I mean, it's not cold to us, right? But it's kind of cold for a lizard in the morning when they haven't had the sun to warm them up for a while. So the sooner that a dominant male can go out there and get his body temperature up to optimal body temperature, he can run faster when he's warm, he can digest food better when he's warm, and most importantly, he can beat up other boys when he's warm. So he wants to get his body temperature up really quickly. So his coloration, we're hypothesizing at this point, is a combination of being really showy with the red head, and it confers thermal benefits, being black on the rest of the body. Now, when you're up in the canopy, remember, think of the umvelt of these guys. When you're up in the canopy, there's lots of really bright light, lots of reflectance. You might be displaying against a really blue sky. The color that you want to be is a really darker color because those colors are more conspicuous. And if you're a dominant male and you want to display to all the other boys that, you know, this is my turf, you want to be conspicuous. So that's, again, a hypothesis of why this male has a red head. Red, orange, and black are the three most conspicuous colors against a blue sky and glary leaves. So there is a question, does dominance come from this coloration, or does coloration lead to dominance? That is an awesome question. For this species, obviously, we don't know, but for other species, coloration is often linked to testosterone. So the coloration and the dominance have a root cause in the level of hormone, which could in part be genetically controlled, and it could be in part environmentally controlled based on how healthy an individual was growing up, how many advantages he had. Yep, exactly. So like the marine iguanas, if you warm more quickly, you're going to be more successful at foraging and at beating up your competitors. Okay. So we're now going to move down from the canopy to the forest floor. Again, remember, back on the forest floor, the umvelt's completely different. The microhabitat's completely different, if you just want to call it microhabitat. And we're going to talk about the bridled forest gecko. Now, the bridled forest gecko, it's actually a dwarf gecko, and it's not really the kind of geckos you think of with the toe pads that kind of climb glass panels. These guys are super tiny. They're like the size of your little finger, and they are weird in so many ways. One kind of horrifying way that they're weird is that one of the defenses for dwarf geckos is that their skin tears off if something grabs them. So you have to be really careful when you hold on to these things, because their skin is just like tissue paper and it'll wipe off, which is absolutely horrifying, let me tell you. But that's normal, that's natural, that's just one of their defenses, same as losing a tail. Okay, so I just told you that red is a really good color for being up in the canopy with when you have full light. Why then does this guy clearly have red stripes? And those are red stripes that are indeed red stripes. We're not, it's not a trick of the eye. Okay, so this guy's got red stripes as contrast, most likely, for his white stripes and his bright yellow stripes. White and yellow are the two key colors that you want to have if you live on the forest floor, because white and yellow are the colors that are going to reflect the best based on the spectrum of light that actually makes it to the forest floor. So these guys, even though they're red, the red is not really the important part other than setting off contrast. It's that white and the yellow that's really exciting. All right, another kind of cool tidbit that I just wanted to share with you. As I was going through these pictures, looking for a nice poster picture for each species, I remembered that when we captured one of these dwarf geckos, I think it was one of the undergrads who caught them, you know, there was some damage to the tail, and we put the lizard in its little drawstring bag, and then when I took the lizard out to measure it, it was in the process of eating the part of its tail that had broken off. Now, most of you are probably aware that tail autotomy is a natural process. Lizards can release their own tails if they feel like they're being threatened. It's an anti-predator defense. So the losing of the tail isn't the interesting part. The autophagy of the tail is the interesting part. And it's something, as I was literally just, looking at this yesterday, it's something that no one has documented in any gecko before, as far as I can tell. Certainly other lizards have documented autophagy, but this is the first record of geckos. So it's kind of cool. I'm gonna try to put out a quick little note in a herpetological journal because I think it's fun. And again, natural history is important, right? Okay. I'd now like to reintroduce you to the blue-lipped tree runner, or the olive tree runner. And he was quite a beauty. He's another medium-sized iguana-like lizard that you might find on the trees, the tree trunks in semi-high light. And this guy looks like he's wearing blue lipstick. And of course, that was definitely the first thing I wanted to study in terms of color for this species. When I took this guy's spectra though, what I found is that my eyes were playing tricks on me. This guy's lips to another lizard were not blue at all, but they were glowing, absolutely glowing in the ultraviolet. So what's interesting here is that this actually got me thinking about all of the other lizards that I've been studying. And it's not only the blue of this guy's lips that are glowing in ultraviolet, but every single lizard I measured in Peru and then later in Central America had glowing UV lips. So it's actually, we all think, we tend to think of ultraviolet as like the glowing colors, like if you go into a club and your shirt glows. But what we actually, what we think that actually it looks like to a lizard is just another color, right? So we see blue, they just see another color called ultraviolet. Our association with ultraviolet's a little different than it actually is, but it's just another color to a lizard. But the cool thing is that now we know that pretty much any lizard's lips are ultraviolet, and that's really cool because of this really flattering picture of me. When lizards are displaying to other lizards, they do this thing called a gape display, which means that they open their mouths really wide as kind of a showy, like, hey, I'm really big, I could bite you really hard display. And that's what this, that's a larger gecko that's doing to me right there, and I gave it right back to him. But in any case, what we're going to look more into in the future is investigating, like, really, is it true that all these lizards that have gape displays also have these ultraviolet colors surrounding their mouth? So it's kind of a cool thing, and we're hoping to look more into it. So we didn't actually put the lizard in the spectrophotometer. The spectrophotometer has a little probe. So it looks like a tiny little micro flashlight. It's about the size of, it's about, I don't know, a quarter of the width of a pencil. And you just press it against the skin. So it's not invasive. You just kind of just touch the little flashlight to the surface that you want to measure. All right. All right, so I know this is not a lizard. I'm not that bad of a scientist. This is a frog. It's one of the poison frogs that one of the guides at Napo brought to me. And just for fun, we decided to measure this one as well. Just like some lizards want to be really conspicuous to their competitors, many other animals want to be conspicuous to their potential predators. So that's what poison frogs do. Their yellow coloration is, hugely standing out in the forest floor, and even their blue coloration on their legs of this particular species is very distinct from the leaves and the other veg that they might be sitting on. So this is called aposematism, and it's a so actually so the spectrophotometer, I'm happy to chat more about how sensitive it needs to be. It has to be sensitive enough that you can measure very small areas of color, because lots of these creatures are very small. But I'd be interested in knowing what kind of vernier device you've got. So hopefully we can chat later. So like I said, these guys are aposematic. They have accumulated poison in their bodies because of the ants that they eat. So basically they are screaming to their predators, don't you dare eat me or you're gonna get really sick. And just as a little fun tidbit, just to remind you all of the difference between poison and venom. If you bite it and you die, it's poison. If it bites you and you die, then it's venom. So there are almost no poisonous snakes. There's actually a couple, but there's almost no poisonous snakes. Most snakes are venomous, and most frogs are poisonous. All right, so now I have two more critters to show you. And they both kind of tell a similar story, but one is much more sophisticated than the other. So the first critter is this Amazon bark anole. And according to pretty much all counts, they're not very interesting. There has been a little bit work done on them, but they're kind of just your standard run of the mill anole. And our recent guide of herps in the Amazon say that they are generally found in the ground or at the edges of clearings. Well, somebody didn't tell this guy that he was only supposed to be on the ground, because there's definitely a solid population up near the canopy walkway of this species, which is something else we didn't know about. Again, just highlighting basic natural history, stuff we don't know about all of these species. And so yeah, they're definitely up hundreds of feet in the air where they, according to the book, should not be. All right, so the other interesting thing about these guys is that they're a really good example of how animals balance being conspicuous to other lizards with being basically avoiding their predators. So anoles, almost all anole species have dewlaps, which they can pull out at will and then retract, kind of as like a flag to wave to their conspecifics, the other lizards of the same species. But what's interesting about this guy is when I captured him, he was a solid kind of brown color, and only after a couple minutes did he change this mottled color. And it seems to me, and I'm still working on figuring out whether we can test this or not, it seems to me that there is rapid color change in this species that directly would make this individual less conspicuous, excuse me, when he's sitting on the barks of these trees. So we even measured the dorsum compared to the lichens that they would be sitting on, and the spectra are very similar. But this is a story, like I said, I don't have too much information on yet. This is another neotropical species, another anole, that I'd like to tell you a little bit more about because we found some of the same things. And I recognize it's getting close to 9 o'clock, so what I'm going to do is I'll tell you about the anoles, this one species, and then I'll wrap up pretty quickly in case there are questions. Okay, so this guy has got a really cool color change. Very dark. Okay. And this happens over the course of maybe a minute, so it does count as rapid color change. So it goes from kind of a chocolate color to a very bright color. Now, the cool thing here is that despite what the media tells you, it's very uncommon actually for lizards to change color rapidly for camouflage. However, that seems to be what's going on with this guy and also the Amazon bark anole. So we wanted to test this, doing an experiment where we tethered lizards in different microhabitats within their habitat, their general habitat, some of them under rocks, some of them on rock walls where there's lots of moss. Now, this kind of experiment is all well and good, but again, we wanted to make sure that our bias as human beings didn't play into this. So what we did is first we made sure that there was no ultraviolet coloration on these guys because that would preclude photography, but we used photography to look at how the coloration on these lizards compared to that of these different backgrounds in their overall habitat. But in order to do this, we have to correct for how their predators, birds, would see them. So first we had to correct for the cone sensitivity of birds. Then we had to correct for bird visual acuity. And then that was a little blurry, so we have to reconstruct those sharp edges. And just wrapping up, what we can see is that using bird visual modeling, we could see how a bird would see anoles on these different backgrounds. And using the things below on the screen, which are called color maps, we can show that coloration does rapidly change to better match the colors of the environment. Okay, there's one more thing I want to show you just for fun. Same species, cool anti-predator behavior, again highlighting the importance of natural history. This species, the water anole, as its name implies, lives near streams, and it jumps into streams when they feel threatened. The species was only described in like 1950 something, so again, not much is known about it. One day we were investigating the anti-predator behavior, and we came across, and I stuck a camera underwater watching what these guys were doing, and this is what we found. What's actually happening is they're rebreathing a bubble of air that is kind of clinging to their bodies like a slick all over their entire body. And they're rebreathing this air, and that's allowing them to stay underwater for a really, really long time. The most I clocked it was 16 minutes, but it might be longer. Okay, so we also ran some lab experiments just to figure out, again, just to show you the process of what we do. How the oxygen tracks in that bubble. So we had a little oxygen microsensor. And what we did find is that over time, oxygen drops. And we also looked at this across phylogenies and found that the aquatic anoles would rebreathe bubbles where the non-aquatic, even though they had the plastron of air stuck to them, they would not. So this, I think, I do wish I had more time. I apologize for perhaps talking too much about my lizards, which I love so much. But I probably won't have time to talk about the social side of the biases, which I do believe is super important. One way that we as Morpho are working to combat this is by increasing diversification in science. And we're working to start research experience for undergraduate program, hopefully, if it gets funded during the next round, that will promote Native students, so Native American and Pacific Islander students in their research at ACTS. So at this point though, I'm gonna just stop, and if there's time, I can answer questions if anybody would like.
Thanks, Lindsey. I guess we'll have to invite you back. Darn it.
Sorry.
To continue this conversation. So folks, if you've got, we do try to be honest with our timing, but Lindsey's, I'm sure, willing to stick around for a little bit and answer questions. Feel free to unmute yourself and go for it. Anybody? Kathy Law's got a question. So why did they emit and then absorb the air bubbles? You were a little hard to hear when the video was playing.
Oh gosh, I'm sorry.
It's all right.
So they're semi-aquatic species, and they jump in the water when there are predators coming nearby. And let me just sneak back. The reason that they're kind of pushing the bubble out and pulling it back in is just because it's part of their normal respiratory cycle. So what they're actually doing is they're rebreathing the same little bubble of air so that they can continuously extract more and more oxygen from it. Because as we all know, even the air that we breathe, when we exhale, there's still plenty more oxygen in that air that we could breathe if needed. So that's apparently what these lizards are doing, allowing them to spend more time underwater. And it's just really cool. It's a cool new natural history finding that we hadn't learned about before, but you know, I'll be happy to share more findings. We just got a paper accepted on this, so I can even share the paper with cool graphics if anybody's interested.
Thanks. Anybody else have questions? Feel free to unmute and just go fire away.
And there's a question here about whether it was clinging to their bodies as well. Yep. So there is a slick of air all over them because their skin is super hydrophobic. So they basically have air that's stuck to their bodies and it gets circulated around as they inhale and exhale.
Wow, that's interesting. I just want to add that I just recently learned that the star mole and a certain type of water shrew also breathes underwater, but that's to taste the water to detect prey, which is fascinating. So these guys aren't doing that. It's not to kind of find their food source. It's just to kind of hold on to. It's almost like you've got like this air, like your aquatic lung, you know, and you're just sort of hanging on for dear life as long as you can underwater. That's fascinating.
Exactly. Cool.
Yeah,
Yeah, it's kind of like the Harry Potter bubblehead charm, if any of you are familiar with that. So that's, you know, it's a bubble that clings to them and allows them to breathe air. So it is like a little artificial external lung.
And I have one more question, if you don't mind. Why are we fooled to see a color blue when it's actually beyond our train of vision? Like, why did you surmise that that's not actually what we saw? That was interesting. Like, we see things that aren't actually there color-wise.
So what we see blue, and our eyes are indeed telling us that is blue. So we are correctly seeing this as blue. If you were another lizard of the species though, or frankly, any other lizard, because most lizards can see in the UV, the fact that this spectrum peaks in the UV means that any blue coloration that we see, the lizard wouldn't even register in their minds, only see the UV color.
And that's it for tonight, everyone. Thanks for joining us. If you'd like to see upcoming webinars, you can find the registration information at morphoinstitute.org/webinars. And you can also access recordings of previous webinars and the curriculum resources that we're putting together to support those. Thanks so much again for joining us, and we'll see you next month.